gene network starts changing. So each of these cells starts expressing or not expressing certain genes, and then they change type. So one of the first decisions that they make is either to become these green trophectoderm cells or to stay inside, like these purple blobs, and are inner cellular mast cells. Then we have another division at another stage. So here the green ones stay around the egg, but the ones inside either become blue or red cells. They have these names, epiblast and endoderms. So we can map what happens in the genes. So we can have like 50 to hundreds of genes, but we can also reduce the dimensionality. And most of the changes happen in a lower dimensional manifold. So as you can see on the right, I reduce the dimensionality with principal components. And you can see that at stage 8, so when we have 8 cells, these yellow dots are all in the same area of the space, so they're indistinguishable again. Then at the blue stage, they are still, again, occupying the same area of the space. But then at stage 32, the red ones have already taken to very separate areas. And so we can see the first division. And then lastly, at stage 64, so 64 cells, we can see that the one on the right-hand side further developed into 2 separate kinds. So again, even with 2 dimensions, we can already see these major changes. So just to give you some motivation, I'm going to first talk about how we can model these setups, and then I'll introduce the inferential methods that we use to estimate these the parameters of these models that we use, and I'll draw some conclusions at the end and talk about what I'm working now. So for the model, we took inspiration from a metaphor in developmental biology. So the Waddington landscapes are these ideal landscapes where each of the cells is seen as a marble going through a developmental path. So it can take certain turns, certain valleys, and when they reach the bottom of the valley, that means that they're fully specified. And the landscape is basically drawn by the gene setup. So we took this idea, which was just a metaphor, and we thought of a potential landscape to which we can associate a gradient system. So originally, stem cells are all the same. So it's like they're sitting on the same attractor, which is that red dot at the center. So here I just drew one of the contour plots. And then we know that in the next stage, they should bifurcate. So we have a pseudonode bifurcation. So all the cells that are sitting at the bottom then end up being in a pseudo. So they will either go on one way or the other of the manifold. So that represents well the bifurcation. And then lastly, we have a further pseudonode bifurcation from this attractor into 2 other setups. So already with this simple example, we can have a modeling like this. So we're going to use some archetypical models in the sense that they simplify a lot what we see in higher dimensions, but they use only 2 dimensions, x and y, and very few parameters to describe all these changes. So for this, as Jack introduced, I'm using catastrophe theory. So simply what I'm saying is that when we have potential functions with 2 dimensions and up to 5 parameters, we can actually classify all the catastrophes, so all the setups of attractors, repellers, and saddle points that we can have. So that's called catastrophe theory, is equivalent to bifurcation studies, really. So you don't have to go through all that I wrote, but it was just to give you an idea of which kind of potential we're using. So a simple one is the elliptic umbilic, when we have 3 parameters that control the 2 dimensions of this setup. So from the potential, we can find the associated gradient system by differentiation, and then we add some noise to the system, just linear noise. And so this should recreate what we observed in the data. The one on the right instead is called double cusp. So I'm using slightly more parameters to be more flexible. And yeah, so these are 2 of the models that I used, and I'll see some example later. So as I said, these are the 2 SDE systems. So you can already think that it's going to be pretty complicated to get a likelihood out of them or a density distribution. So what happens when we don't have a tractable likelihood is that we use likelihood-free inference methods, and they divide mostly into categories. So one is using an approximation of the likelihood, and the other one is approximate Bayesian computation. So first, the method that we develop is in the context of surrogate likelihoods. So we want an approximation of it. Now, the first approximation that we make comes from the theory of linear noise approximation. So the main assumption is that given our multidimensional system, our SDE, this can be decomposed in a deterministic solution, and then we consider some fluctuations that are the noise. So this translates into basically thinking of a system that is driven by a Gaussian distribution, where the mean follows the deterministic solution of the system, and the variance account for the level of fluctuation that we look at. So it's an approximation because we basically Taylor expand but stop at the first order of the noise. So this is quite nice because the only thing that we need is the mean and variance of this Gaussian that can be obtained analytically as an ODE system from the original one. And it works pretty well as an approximation. So here you see some Euler-Maruyama simulations, and the ellipse, the blue one, is the corresponding Gaussian that is developed. So it covers up the data pretty well, and it's really fast to compute. And it's a Gaussian, so the likelihood is available. However, the problem with this is that it doesn't work as well for saddles. So here there's another simulation where I start on the stable manifold. So at some point I'm going to hit the saddle, and what happens is that the Gaussian has a variance that elongates and blows up, because basically the fluctuations that would be negligible before are not anymore. So either you expand the orders that you're looking at for the approximation, but then it's very time consuming, so why are we doing that? Or you do a further approximation. So here we develop the Fokker-Planck LNA in the sense that we use the standard LNA with one Gaussian until a critical point. And then we stopped the LNA, we projected the density from 2D to 1D. And then we just looked at the projection on the manifold, which is where the main changes of the dynamics happen. And so this, when we stop the LNA, we use this as the initial condition of the Fokker-Planck, and then we iterate using the dynamics until equilibrium. So then we have a 1D expression of the density. So each of the data that we observe, we can map them into one dimension and get a pseudo likelihood of them. So we have two approximations here. So once we have this approximation, we embed it into an inference procedure. So here I use PMC, which is population Monte Carlo. I can tell you more about this if you want, but essentially I'm trying to improve on my prior beliefs on the parameters through stages of simulations driven by the pseudo likelihood. So the convergence results come from important sampling theory. So here I'm just plotting two of the parameters that I'm looking at. So this is the elliptic umbilic with 3 parameters. And the relationship between U and V is quite good. So these are the area of high density, and I only reconstructed this with 10 data points. However, I need to limit myself in setups where I have only bistable cases. So I only have 2 attractors, not more. So actually I fixed one of the parameters and only try to estimate U and V. So starting from some prior beliefs, quite wide ranges for both parameters, I can actually end up with a quite concentrated density. So my posteriors point quite well at the true values that I use for the simulation. So the green lines are the true values, and here the variance is in the order of 0.2, 0.3. So all my posterior means are quite well reconstructed. And even the cross correlation is quite good. But as I said, that this is only for bistable sets. So if I want to reconstruct a tristable or like more attractor setups, then this theory is not really developed. So we actually looked at another approach, which, as I said before, was approximate Bayesian computation. So the problem is still that with likelihood-free inference, either you approximate the density, the likelihood, or if the likelihood is intractable, but it's easy to simulate from your model, what you can do is, okay, I use some parameters for my model, I simulate from data from there. So ideally, I want to find parameters such that my simulations are quite good with respect to the data that I observe. So in order to improve on my parameters, what I do is I try to compare simulated and observed data. And so for doing that, I need some discrepancy measure. So if you do some epidemiology, what you do is, for example, you compare the number of infected people from your model and the observed ones. You take a difference and then you improve on that difference. So you know that when your model is perfect, you should have 0 difference or something around those lines. And then that creates a divergence. So it creates a way for your model to improve on this. Now, the problem with our cases instead is that we have 2-dimensional data. So how do you compare them? So basically the idea was focusing on discrepancy measure that could do that. So sorry for the heavy slide. I just wanted to give you all information, but essentially, the first discrepancy measure that we looked at is the Wasserstein distance. So the Wasserstein has been developed in optimal transport. So this is the analytical form. It's basically a way to quantify the distance between 2 probability functions. So if my data come from 2 different probabilities, then empirically I want to compare the 2 densities, and I want these 2 densities to be really close to each other, actually the same one. So with real data, this is the computation of the expression. But the interpretation is the following. So if you have n observed data and m simulated one, what you can do is take the L2, the Euclidean distance between all these pair of points, and you want each of these points to correspond to one from the other distance. So this is actually an optimization problem and can be solved in a closed form with the Hungarian algorithm when you have equal numbers of data. So you compute the distances between all of them, and then you associate the closest point to the one in the other data set at the cost of n3. So if n is your numerosity, it's quite expensive. So there's also a regularization version that is the sequence algorithm, but here we use the exact one. So this was one of the measures that we looked at. The other one comes from probabilistic forecasting. So in probabilistic forecasting, you use scoring rules, which assess how well you're predicting your observed data Y using the distribution P. So when you have a value for the scoring rule, it's like saying that how am I penalized when I predict Y using P? So obviously, if my P, my distribution is the real one, then I shouldn't be penalized. So it should have a minimum when I use the true distribution. And this is called propriety, or like when a scoring rule is proper, then this is the minimum value. So if a scoring rule is proper, then I also can define a divergence. And so the divergence can be minimized with respect to my ideal distribution. So there can be many scoring rules. Some of them are not proper. In this case, obviously, we considered a proper one, which is the energy score. So again, I'm just giving you the formula, but again, the scoring rule for the energy score as a divergence, which is the square energy distance. And it can also be estimated empirically in an unbiased way. So again, this is the empirical version that we use. So ideally, if you generate X data from P theta, so from a distribution using the theta parameters, then when I compare them to the observed Ys, I want to minimize the scoring rule, so the average scoring rule, and that minimum should give me the ideal parameters for my setup. So results. This is running the population Monte Carlo ABC, so similar setup to that one, but to the previous one, but we use ABC measures. So using 1,000 particles with 7 steps, we compare the results for Wasserstein and energy score for the elliptic umbilic. So here we don't have to restrain ourselves to bistable setups or so on. We can look at all 3 parameters and the noise together. So we can see that both posterior distribution, these are the marginals, are quite concentrated around the true value. And yeah, so the means, the posterior means are quite good. The Wasserstein takes a bit more to estimate one of the parameters, but it's a pretty good result. So we also did this for a tri-stable case, so when we had 3 attractors. So we started with some observed time points in green. So it's a mixture of Gaussian centered at the 2 saddles. And then we observed them at the end. So when they are already clustered in the 3 points. And even in this case, the posterior estimates are really good. So just because I mentioned before that the scoring rule was taking a bit more to converge, I also used 10,000 particles. And again, in that case, obviously it improved the posterior distributions. So we went even further. So as I said before, for the elliptic umbilic, I was just looking at one snapshot. So how do I pass from one attractor to 2 or from 2 to 3? So what if I look at a sequential setup? So when I observe all the 3 transitions. So what I mean by that is that I start at a saddle point, I observe the red data. Yeah, well, okay, conclusion is, yeah, we try for a sequential setup for those of you in the room. So also these results are quite good. We investigated on some of the identifiabilities that we couldn't estimate well, and that made sense with the values, like the geometry of the landscapes. So in the future, like, yeah, so I was just summarizing some of the comparisons. So obviously the ABC works. The Fokker-Planck worked well. It was really concentrated. It works with very few data points. The ABC, though, generalizes even more. So we could use it for all kinds of setup we're trying to recreate. So it's easy to generalize. The Basini is a lot more accurate, but the scoring rule is cheaper. So it's just about balancing how many particles you're using in your algorithm. So yeah, this concludes one part of my research. Now I'm thinking of doing some future work where I don't have a fully parameterized potential. So instead of having the closed form for V, I want to learn it with a neural network and see if it can be more flexible in recreating some of the more complicated setups. So that's what we're doing at the moment. And thank you for listening. Do you have any questions?